Today's plenary session is about clinical trial readiness. I'm sure many of you have heard this term over the course of your journey with a muscle condition, but what does that really mean, right? I'm pleased to welcome Dr. Reghan Foley, Dr. Carsten Bönnemann from the National Institutes of Health, Dr. Alan Beggs from the Boston Children's Hospital, and Dr. Gustavo Dziewczapolski from Cure CMD to walk us through this topic.
So as Rachel said, we're going to be discussing clinical trial readiness, and I'm joined by Dr. Carsten Bönnemann, Dr. Beggs, and Dr. Dziewczapolski. And if you have questions, you can include them in the chat, and we'll try to address them as we go along, if we can. So what does clinical trial readiness mean? It means being prepared for clinical trials that are in preparation. And to prepare for clinical trial readiness, there's really three main elements. The first is optimizing clinical care. The second is identifying outcome measures. And the third is studying the natural history of the particular congenital muscle disease subtype. So optimizing clinical care enables everyone who's enrolling in a clinical trial to start from the same optimal point. And this is essential. And we've learned from prior clinical trials that not having the optimized clinical care can complicate the data of the trial and perhaps negatively affect the ability of the trial to capture a positive outcome. And so we've learned to kind of now focus on ensuring that all patients begin the study with good optimized clinical care. And we talk about proactive care. In particular, proactive respiratory care is a really important element for all forms of congenital muscle disease. And typically the lung pattern you see on your pulmonary function is described as just restrictive, quote unquote. And this is because there's restriction of lung expansion due to weakness of the intercostal muscles, weakness of the diaphragm, and some decreased compliance or stretching of the chest wall. So this pattern really is indicating extrinsic causes of restrictive lung disease. The lungs themselves are fine. It's the muscle weakness and the stiffness of the chest wall which affect the pulmonary function. And because of this pattern that we see within all the subtypes of the congenital muscle diseases, we have an approach of proactive respiratory care, and that includes pulmonary function testing in both the upright or seated position and the supine or lying down position. Doing this in both the upright and supine position can capture diaphragm weakness and is important in terms of determining if and when non-invasive ventilation should be begun or should be started or initiated. And polysomnogram or overnight sleep study is the gold standard for assessing hyperventilation or during deep sleep or retaining of carbon dioxide during deep sleep. And so I recommend frequent pulmonary function testing and a polysomnogram to initiate non-invasive ventilation, also to confirm that the settings of non-invasive ventilation are adequate. So with that approach, our patients then often have a timely initiation of bi-level positive airway pressure and non-invasive ventilation when needed. And that's the ideal standard so that we are being proactive in providing the adequate clinical care and not reactive in starting things after they're already needed. We also recommend the use of the insufflator, exsufflator, or cough assist machine to promote coughing and airway clearance and promote expansion of the lungs and chest wall. Proactive cardiac care is also important for congenital muscle disease and should include echocardiogram or ultrasound of the heart, EKG, and Holter monitoring, which is prolonged EKG monitoring. Most subtypes of congenital muscle disease should have both echocardiogram and EKG monitoring, but I wanted to highlight that for LMNA congenital muscular dystrophy, careful monitoring of arrhythmias is absolutely essential with frequent Holter monitoring, and based on the Holter monitoring findings, the timely placement of implantable cardioverter defibrillator or ICD. So a very proactive need for Holter monitoring and the placement of ICD in a timely fashion when the Holter monitoring findings indicate the need for ICD. For tight-related myopathy patients, careful monitoring of cardiac function via echocardiograms and cardiac MRI for those who can perform a cardiac MRI is helpful for detecting any change in cardiac function. Orthopedic surveillance and care is also an important part of optimizing clinical care, and many patients are assessed for ways of stabilizing or approaches for curvature or scoliosis, and a lot of centers had recommended bracing, and we wanted to always highlight that it's important that bracing for scoliosis should be approached very carefully, because moving the chest wall is very important for respiration and ventilation. So if there is an ability for a brace that actually leaves a space, either like a hard brace or a casting that would leave a space for chest wall expansion, that would be preferable. When surgery is indicated based on the degree of spinal curvature and the rate of progression, it's very important to have good coordination between orthopedics, anesthesia, intensive care, and pulmonary teams to ensure a very smooth preoperative, intraoperative, and postoperative experience. These are all very important to make sure our clinical care is optimized so that when a clinical trial begins, you're ready to start the clinical trial without having to address first these clinical care issues. Cure CMD has many resources available online. There's a recently put together excellent CMD pulmonary guide. There is a management of congenital muscular dystrophy guides, also management of congenital myopathies guide, management of limb-girdle muscular dystrophies, all available under care guidelines on the Cure CMD webpage. And I just wanted to pause here for comments from Dr. Bönnemann and Dr. Beggs about also the importance of registry data.
Yeah, let me thank you, Dr. Foley. I wanted to just kind of reiterate the importance of this theme of proactive and optimal medical care for several reasons. One is that proactive and better medical care actually fundamentally changes what we call the natural history of these disorders. So there is a natural history that is known in the older literature, which was basically a wait and see natural history, where you didn't intervene, didn't do anything, and just kind of let the disease take its course. That is absolutely no longer acceptable. And that natural history was, of course, devastating and really discouraging natural history. But with the introduction of proactive, rationally based and interventional medical care, including respiratory support, cardiac support, this quote-unquote natural history has changed, I can only say dramatically, for many of the subtypes of our conditions. We're not talking years, we're talking decades of different medical progression here. So that really reiterates that we're talking about fancy drugs that we want to find and introduce. But the simple but effective introduction of optimal medical care has made a really astounding difference in the natural history of these conditions. What that also means is that your outcome measures and what you want to achieve in a clinical trial changes. Your starting point changes, your outcome measures change. If you think about an outcome measure that's respiratory, say the dependence on ventilatory support or the introduction of ventilation to a patient, you have to be all on the same playing field with the medical care that is given anyways to a patient, so that you can see a difference that an additional therapeutic intervention actually makes. And you want to avoid that only with entering a clinical trial, a patient will get optimal medical care. The optimal medical care should be there before a trial is even considered, so that all the outcome measures are really starting from the same playing field. So can't be overemphasized how important.
Perhaps I could just give an example relevant to our Selenon community. And as many of you folks know, one of the characteristic features of Selenon myopathy is spinal rigidity and then an evolving scoliosis or curvature of the spine. And clearly, if nothing's done, this can become quite debilitating. It can make it difficult for you to take a full breath. But orthopedic management, initially with bracing and then eventually with surgery, can make a tremendous difference in keeping that spine straight and help prevent further deterioration in someone's ability to breathe and maintain a straight posture. This is of particular interest to Selenon, but in many of the muscular dystrophies, once you become wheelchair bound, this is also an issue. Regarding Carsten's comment about how this affects outcome measures. Of course, if you don't do any spinal surgery, but you do an early treatment, then one outcome measure might be, for example, measuring how rapidly people's spine start to become curved. However, that's not something we would want to measure in a clinical trial because, as Dr. Foley and Dr. Bönnemann have been saying, we want to use whatever medical management is available first to prevent these things and then overlay on that our newer molecular and genetic therapies.
So to get everyone recognized and on the same plane, registry play a really, really important role. And our goal, of course, should be that every patient in our subtypes is part of a registry, recognized there and also followed there. Alan, do you want to talk a little bit about the importance of registries at this point?
Sorry, did you say Alan?
Yeah.
Yes. So, there's lots of different types of registries. There are simple contact registries where, all we have is somebody's name and address and permission to reach out to them. There are research studies, such as the ones conducted by many of us, which actually involve more than that collection of information and contribution to natural history studies, which Dr. Foley will speak about in a few minutes. But essentially, they're extremely powerful and critical and important. I mean, for example, although in academics, we are highly motivated to work with you to develop treatments, it takes the resources of industry to really turn those treatments into something that's medically qualified and proven and made available to patients. And you can't convince industry to do that if you can't tell them that there's enough people out there, there's enough unmet medical need, and that we can reach out to these communities in order to actually enroll in trials and then offer treatments once they become available. So they're really critical. Can't say enough how important that is. The other point I'd make is that register early and register often. There's nothing unique and exclusive about any one. So maybe Dr. Dziewczapolski could talk about the CMDIR in a minute. But I would say for anybody being approached about enrolling in a registry, don't hesitate just because you've enrolled in a different one. What is important and helpful to all of us is to be able to identify people and identify overlap. Because if we want to identify a group of people who might benefit from a therapy, we need to know if we're double counting. So giving permission to share some information in order to identify people that are enrolled in more than one place is great, although it's certainly optional. But just making yourselves known is really very important. Gustavo, do you want to say a word about the CMDIR?
Yes, just that you mentioned about different registries is important, and that's why why CMDIR changed the platform. Now in the new platform, we have the possibility to have unique identifiers so that we can check with other registries if that person is registered in more than one. So that's why we are most of the people in the audience have received or will receive messages about claiming the profile and complete it again in the new platform. It's very important to have, and it's also a good resource to keep your clinical data all together in one place, and also for us to help in knowing your exact mutation. As we talked yesterday, the importance of having an accurate diagnosis, and the registries can help guide you and help you find that answer.
Great, thank you. And just kind of one final comment would be, as we all are very excited for upcoming therapies and trials, I think it's important to recognize that care is essential, and the word care and the word cure are just one letter different. And until we have that therapy ready to be trialed, care is essential. So until we have the cure ready, we need to make sure we optimize that care. So identifying outcome measures is also an essential element of clinical trial readiness. And we need to really have outcome measures that are really appropriate to the age of the patient and also the disease stage, and outcome measures which are reliable outcome measures for really capturing the condition and reproducible on various iterations of repeating the outcome measure, and also measure something meaningful to the patient, a meaningful change, not just a number on a scale, but something meaningful clinically, and most importantly also sensitive to capturing that change. So here is just an example. So we have what we'd say kind of a toolbox, or an array of tools for measuring outcomes in the congenital muscle diseases. And I listed some examples just to give you an idea of how we can capture function. And under the category of motor function, we frequently in congenital muscle diseases use two different motor scales. The motor function measure 32 for age 6 years and older, MFM 32, or the motor function measure 20, MFM 20 for ages 2 to 5 years. And this scale has been found to be reliable and reproducible and has been validated in many congenital muscle disease subtypes. We also use myometry to measure the strength of particular muscles, and we've used that in some of our natural history studies. In terms of examples of outcome measures for cardiac function, echocardiogram, EKG and Holter monitoring, and cardiac MRI, or pulmonary function testing, and also means of measuring carbon dioxide, whether it be via capnography checked like in a pulmonary function lab, a measure of carbon dioxide, or carbon dioxide measured during a sleep study. That would likely be an important outcome measure in many studies that are going to be looking at pulmonary function. And very importantly, quality of life outcome measures, indicating to how your life is affected and how it may or may not relate to your motor function at the time. And I wanted to just pause here in case the rest of my panel wants to make any comments.
Well, I'll just say there's a whole industry around identifying the correct outcome measures, and for every disease, you really need to specialize what you're going to measure that's relevant for that group of people. And for some of the conditions we talk about here, there's variability. So, in some cases, individuals may be very weak and not survive much past childhood, in which case you need to measure things early on. In other cases, people walk, and you need to measure aspects of how well they're breathing or muscle strength in different muscle groups. And so oftentimes, a lot of these measures are optimized. So the MFM 32 or MFM 20 that Reghan just mentioned can be very useful for patients with some conditions, but maybe not so much for others. Or if you're applying them to a disease where they haven't been validated, you need to do a study to just demonstrate that they actually measure something. And then finally, we want to think about how these measures actually correlate to quality of life and to things that really matter to people. So, can you raise your hand to your mouth to scratch your nose when it itches? That's something very important. But maybe some esoteric measure of muscle contraction may not matter that much to a person. So we need to think about how these measures correlate to somebody's ability to live their lives in a good way.
Yeah, outcome measures are they're a complicated dance, if you will, that we have to go through and have to do in order to develop therapeutics. And similar to what we discussed yesterday in the toolbox for gene-directed therapies, I think the more we can toolbox this, the better it's and the more efficient it's going to be. And for several reasons. One is if an outcome measure has been validated and found to be useful in a disease that is not ours but related in functional aspects, then we can port that over to the disease under question and validate it in a relatively stringent and short period of time, just to show that it's equally applicable to the disease that's now being studied. But the more we use outcome measures that are validated and found to be useful elsewhere are the more efficient we can be in the outcome measure development. And then we also know that people have familiarity with the outcome measures, such as the regulators that we want to address in the end, and also the evaluators at the different centers where we want to do our study. And the second layer of complexity is really from biology to function. So these our conditions that we're dealing with here are not exclusively, but for the most part, disorders of skeletal muscle, and then different layers of other systems on top of that, such as cardiac muscle and also brain function, for instance. But in the end, we want to have an outcome measure that really reflects that our therapy works where the disease is biologically happening. And that can be at different levels of granularity. You can go into the molecular sphere, and we talk about biomarkers later, and show something that the therapy is supposed to change in the muscle is actually changing. But then we have to go to the next level and say, is that what we're changing on the molecular level changing the function of the muscle? So we need an outcome measure that measures how the function of the muscle works. So those could be strength measurements or motor function measurements. And then we have to go to the next level and say, is that functional change in the muscle actually making a difference in the patient's life? So then you come to that quality of life and functional level. And they have to be interlinked, and they have to be convincingly interlinked. And when they're convincingly interlinked, sometime in the future, we can go then to a level that is a bit more granular and say, well, we're moving this biomarker. Therefore, we expect that we're also going to move the function of the patient and make our trials much more efficient and quick if we can show that link. But we have to show that link before. And so in our outcome measures, we have to be cognizant of all these levels of outcome measure, the molecular, the functional on the muscle level, and the functional on the patient level, if that makes any sense. But this is what's going to drive our outcome measure development. So the applicability of toolbox approaches, and then also the link from biology to function in our therapeutic approaches. In the end, we have to go through all of that because we want to convince ourselves, you, the patient community, and the regulators that a therapy that we are developing actually is working or maybe not working. And then we should kind of finish that development right there and move to the next therapeutic option. That's why this is technical, but important.
And I believe Cure CMD has posted on the conference portal a resource, educational resource also regarding outcome measures. Just to give you a quick example, because this is a kind of information that you'll be hearing when you get involved in either a natural history study or a clinical trial, there are multiple motor scales that are available for assessing motor function. In particular, I wanted to highlight for early motor function, there's been many different motor scales that have been used in various neuromuscular conditions and have been validated. And you'll probably hear your physical therapists using these terms. Obviously, we mentioned the motor function measured 20, but also, but also there are other scales such as the CHOP-10, which is the Children's Hospital of Philadelphia Infant Test of Neuromuscular Disorders. So typically the physical therapist will use a scale which has been validated in that age range. And over time, there have been efforts to also use a scale that may be able to be applicable to multiple ages, such as this new scale, which has been developed by our colleagues at Columbus, at Nationwide Children's Hospital in Columbus, Ohio. They call the Neuromuscular Gross Motor Outcome Scale, which actually then enables, which enables the same scale to be used from birth through 5 years of age. So then kind of making a more seamless transition as the child ages. And hopefully this will be an outcome measure that would be able to be used for our patients and then be applicable through those the different stages and still capture the excellent motor function that's captured within the CHOP-10 scale. And this also combines a scale, which is the Hammersmith Functional Motor Scale Expanded. Again, these are all scales that have been validated in early onset or a congenital muscle disease. So we had done at the NIH a CMD Common Outcome Measures study. It was funded by Cure CMD and the NIH, and many of you participated in the study. And it occurred over 4 years duration. So it was 5 summers in a row, but it was like a 4 years duration study looking between ages 4 to 22 years at collagen 6 patients and limit 2 patients. And what I wanted to highlight was what we learned from the study were many things, but one thing I wanted to highlight was using the annual rate of change in the MFM32 total score among the patients with collagen 6 related dystrophy. That was found to be a change of 4.01 per year. We were able to use that to then perform a sample size calculation to detect basically, if we wanted to power a clinical trial or study with 80% power, detect a change in 12 months of the forced vital capacity. We were able to calculate that we would only need 12 patients to detect a change of 4, meaning a stabilization of disease. So if the therapy were able to stabilize the pulmonary function and prevent a decrease, 12 patients would be adequate to prove that. So again, these outcome measure studies are important for validating outcome measures, but also for preparing for clinical trials, for figuring out, the size of the sample size needed to prove that a therapy would prove to regulatory agencies that a therapy is indeed an effective therapy. Oops. And then this is kind of just a schematic summarizing the data that we found from this clinical outcome measure study in COL6 and LAMA2 related dystrophies. And that is that the MFM32 was very powerful in capturing change in function and correlated with a lot of different measures. But one point I want to make here is you will notice that the quality of life measure did not correlate with changes that were captured in other outcome measures. And this is a very interesting phenomenon which we see in our muscle patients. And I don't know, Dr. Bönnemann, if you wanted to comment on this.
Yeah, it is the kind of quality of life paradox that's now known not only for early onset muscle disease, but also for other severe genetic disorders, mostly of childhood, which is that the quality of life scales underestimate the resilience and positivity of the patient population. Meaning that when an outside observer looks at the child and says, well, you should feel miserable, and the child says, no, I'm actually very good, I'm fine, I'm living a fully fulfilled life, and I'm happy, then the quality of life measurement will not capture that the child may be using a wheelchair, may be needing upper extremity assist devices, but is happy. And so we are which is, of course, for us as caretakers of families with congenital muscle disease are well aware of, and we just admire our patient populations for that. But it is not obvious to the quote-unquote regulators that this is actually the case. And so the quality of life measures really fail in that aspect. They fail in a good way because of the resilience of our patient population, but they're not. We have to disengage the regulatory view from the quality of life view as it is measured currently and reflected in these scales. Now that's different from measuring a change. So of course, families and affected individuals are very cognizant of change in their function, and they can very accurately report whether their function, their abilities have changed over a year, for instance. They haven't changed, or it has gotten worse, or it has gotten better. And that's the change that an affected individual, an affected family will report to the caretakers. And that can then be correlated to the functional outcome measures that are measured at the same time. And we call that minimal clinically important difference. So that's not necessarily a quality of life measurement. It's a measurement whether a functional change has been recognized, felt, experienced by a family or a child or an affected individual. So that's the measurement that we try to correlate with our outcome measures to see what change in our functional outcome measures is actually noticed. It is actually taken into account by an affected individual and say, well, my disease has gotten worse, or has gotten better, it hasn't changed.
So here we have the, as Dr. Bönnemann just mentioned, the minimal clinically important difference. And so determining the clinical importance of any given change that's captured by outcome score. And so this is the paper that we had put together in collaboration with our wonderful colleagues in Lyon, France, and using the same exact data from the CMD Common Outcome Measures study, they correlated the MFM32 data with self-reported impressions of change in function, and were able to then define the minimal clinically important difference for the MFM32 scale, either 2.5 or 3.9, depending on the analysis. But this, again, is very important data to present to regulatory agencies that when you're going to begin a clinical trial, you not only know how to capture that change, but also have anchored that change to a change which is important to the patient.
And you can see there in the for the MFM32, really across many neuromuscular disorders, really it always hovers around 4% about. And so you saw in our natural history study for the COL6 related dystrophies, there was a change of about 4% over a year. So that's a change that is recognized. And if a clinical trial were to change that to no change or to a positive change over a year, that would be clinically meaningful.
And I just wanted to briefly touch on exploratory outcome measures. They're exploratory because they're not yet validated, but yet they are still outcome measures which we are studying because they may also adequately capture function or change in function over time. And so we often want to avoid doing multiple muscle biopsies during a clinical trial to prove a change or to prove engagement of a target, meaning the muscle. So we've looked at muscle imaging, different biomarkers, and also the potential of remote outcome measures or capturing motor function while you're still at home. So just to touch on this briefly, muscle imaging, obviously for use in a clinical trial, ideally you want to use quantitative type of muscle imaging. And there are different ways to study the MRI data that can quantify fat replacement or looking at the chemical composition of the muscle. And these approaches have been used in different subtypes of muscle disease, in particular in Duchenne muscular dystrophy, looking at the quantitative MRI of fat replacement. Actually, in patients with Duchenne muscular dystrophy, is found to be more sensitive in testing and capturing clinical change than perhaps some of the motor scales. And so we've already begun to look at the potential of using quantitative MRI in our patient populations and have captured data. In particular, we have so far data in our collagen 6 Lambda-2 dystrophy patients and looking at different exploratory means of change in the quantitative appearance with T2 and MRS fat fraction over time. The complicated element here is lying still for prolonged MRI scanning, which often is the case for a quantitative MRI. It's very challenging in individuals that need non-invasive ventilation or may be uncomfortable. And so trying to get a short sequence, a short MRI scan that's still quantitative has been our goal and something we're still working on. But again, it could be a powerful biomarker of change of the muscle, which is non-invasive. Just to give you a quick example, if you perform muscle ultrasound in a child, this is from a young child, 9 months of age, with Lambda-2 dystrophy, the muscle ultrasound is already quite, you kind of see appearance we call echogenic, or kind of a increased white appearing in the muscle, so unable to really be an adequate biomarker because it's kind of maxed out. It's a very sensitive muscle imaging modality. However, if you look at muscle MRI, here's a muscle MRI in a child of 8 years of age with Lambda-2 dystrophy, and you can see that it very well captures the muscle appearance and also the involvement of certain muscles more than others, and would be a more adequate means of potentially capturing quantitative changes over time in the muscle appearance. And to briefly touch on the concept of biomarker is a very important concept in designing clinical trials and our clinical trial preparation. Biomarkers are markers that are disease relevant and ideally capable of reflecting a potential therapeutic change or change that the therapeutic compound has resulted in, and ideally would provide a measure of disease and target engagement. So trying to see if something that's intrinsic to the pathophysiological process, the underlying process of the condition, has that changed because of the therapy. So there are different means to evaluate biomarkers, and this work still remains largely exploratory because we've we have to always pool and collaborate with all of our colleagues on the data that's been collected to see what may be the most viable and reliable biomarker. But typically we look in serum or compound or looking at the blood profile, urine, or muscle tissue. We have some colleagues who've done proteomic studies or studies of protein expression in the muscle tissue. Again, an important element for identifying perhaps a means of changing the condition without being too invasive. And again, muscle tissue would require a biopsy, but if we can find a corollary in the blood or the urine that could be changed with the therapy, that would be very helpful.
On imaging for that matter, because muscle imaging, quantitative muscle imaging is a biomarker in its own right. This comes back to what I mentioned earlier about the different levels of therapeutic target engagement, if you will, from the molecular level to the functional level on the muscle to the functional level on the patient level. So the holy grail for a biomarker is to reflect a functional difference or not. So if you have a biomarker that's very sensitive and changes into a certain direction before the functional level on the muscle or patient level changes, then that is a very powerful and strong a link between target engagement and eventual functional outcome. You have to show that. You have to prove that. But once you've proven it, you can actually use the biomarker as a surrogate endpoint, as an earlier endpoint, as a sensitive endpoint to make decisions about your trial. And so if you know a biomarker has to be moved in a certain direction to be effective on a functional level later, and you don't see that biomarker to move at all, then you can maybe declare futility for a trial earlier. Or vice versa, when you see a biomarker work in the right direction early on, then you'll put much more effort into developing that particular therapy into proof of efficacy trial, and put more resources and more patient effort into that. So biomarkers are difficult, they're hard to prove, they're hard to get at, they're really tough, but they really are an essential thing to keep looking for because it would make our trial so much more efficient. Ellen, I think, wanted to comment on that as well.
Yeah, I mean, I'll just say, as Carsten just said, they're very powerful if we can identify them. And so I just want to take this opportunity to put in a plug for community participation in the research. And so many of you who have enrolled in some of our research studies have been asked to submit samples of blood or maybe leftover muscle from a diagnostic biopsy or collected during a surgery for other reasons. And these are extremely powerful and useful. So, we store these things up. We keep them on behalf of the participants, are able to ship them to other researchers on request if that becomes a useful thing to do. And in particular, Cure CMD has a tissue repository based at the Medical College of Wisconsin, and I think many of you have contributed to that. So understanding the value of these very precious samples, especially when we're talking about rare muscle diseases, we just can't overestimate this value. And so thanks to everybody for their participation, and hope you all understand the tremendous value it brings to making progress in these diseases.
Just to briefly mention, so again, exploratory, we're looking into various means of remote monitoring to gather motor data while you're in your home setting, your natural environment, which could be a good corollary to the formalized assessments performed at your hospital running the clinical trial. So we looked into different wearable devices that may be helpful in capturing function in the different congenital muscle disease subtypes. Obviously, we need to assess these devices for the feasibility of wearing. Are they comfortable? Can they stay on? Are they too heavy? Or is it feasible to keep on all the time for a few days duration? And the ability to reliably measure a meaningful change in motor function. So again, currently exploratory, but maybe implemented and has a great potential. So moving on to our last category, that of studying natural history. And this is so important so clinical trials can be powered with lower numbers of patients and can capture disease course and capture changes. So these are really kind of the best means for us to prepare our clinical trial design to capture change. And again, ideally with lower numbers of patients. So I wanted to highlight two recent retrospective natural history studies, meaning studies that have gathered data from the past and put that data together. And also to highlight that often registries can also serve as a means for capturing this type of data as well. This study was done as a very large international collaboration and gathered data on 151 patients. And it's been very important to gather this data in the LMNA-related CMD community because it is a rare condition. So we need to really, come together across the globe to put our data together. And I think what's been essential is in highlighting that, yes, there is a great variety, a great spectrum of presentations in LMNA-related CMD. However, we were able to recognize that patients that had early cardiac interventions or the early, involvement of abnormal cardiac rhythm seen or early cardiac medications, those patients typically had early or pulmonary interventions. And we saw from the data here that the median age for initial cardiac rhythm abnormality is 11 years of age, while the median age for initiation of nocturnal non-invasive ventilation is 16 years of age. I don't know if you want to make a comment, Dr. Bönnemann.
No, not at this. But we can make a comment on the value of retrospective.
Perfect. So just to summarize, this study was done by our colleagues in London. I think they're actually probably on the call today. So Dr. Alberto Zambon and Dr. Anna Sarkozy led this great effort to gather data in their large cohort of Lambert-Eaton dystrophy patients. So 46 patients, 42 with complete deficiency and 4 with partial deficiency. And very importantly, they were able to gather the annual decline in forced vital capacity, which they calculated was like 2.9% per year. And then they used that data to then calculate sample size, and because knowing that forced vital capacity could serve as an important clinical trial outcome. And they basically have demonstrated that if you want to normalize or stabilize that decline of 2.9% per year, you would just need 13 patients. And this is very similar to the data that we actually gathered also from our CMD COM study that recommended 12 patients. So again, giving us a good idea for how we can set ourselves up for success. If we're going to test a therapeutic, can we successfully capture if it does positively affect the condition? And then just to mention, we are also working on a prospective. So going forward, gathering data in the young patients with Lambert-Eaton-related dystrophy, because we don't have that data. And we know that we have good data after age 5 years, but not as much good data between birth and 5 years. And it's important to gather that. So we're going to begin this. We are beginning to gather this data and collaborate internationally with multiple colleagues across the globe so we can harmonize our data collection and use it as a powerful means of setting up our clinical trials as therapies are in development. And then just to mention, this is a very large global collaboration, not only amongst academic medical centers, clinicians, researchers, but also with patient organizations and also with biopharma. So really kind of an example of a large consortium to help pull together that data in a very collaborative means. And then just briefly, many of you know we have also collected data, retrospective data in the collagen 6-related dystrophies, looking at the natural history of pulmonary function. This data as well has been very instrumental in helping us understand what the annual rate of decline is per subtype of collagen 6-related dystrophy. And very importantly, when we dive even deeper, we see between age 5 and 15 years, is actually a steeper decline in that age range. So we have the overall rate of decline per year, but within that age range of 5 to 15 years, the decline is a little bit steeper for the older patients and actually a little bit milder for the intermediate patients who decline more after age 15 years. So again, understanding that the dynamics of the decline and understanding the expected natural history will help us in setting up a clinical trial to be able to measure potential change of a therapeutic. And then just very briefly, similar data in Lambert-2 dystrophies that was retrospective, demonstrating the annual rate of decline and setting us up for having that information for future clinical trials. And I just wanted to set one example here for selenoid dystrophy, and obviously let Dr. Begg speak. This is a study from 2011 of 41 patients, but the point I wanted to make here is the annual rate of decline is not that much. It's 1.1% in forced vital capacity per year. However, by 5 years of age, the patients already had a forced vital capacity, which was on the lower end. And so it kind of highlights the need for non-invasive ventilation as young as age 1 year. But interestingly, that really is because the disproportionate involvement of the diaphragm. Most of these patients remained ambulant, even though they were on non-invasive ventilation.
Maybe I can, because I think Rachel is signaling we're running out of time. I just want to make one last pitch on the power of retrospective natural history data collection through registries, to outreach at centers, at single centers, at multi-centers, because they really help us to plan a prospective natural history study with the outcome measures that we have selected, because prospective natural history studies are very time and labor intensive, and you want to really keep them at the most informative and most efficient level. And the retrospective data collection really helps you identify which outcome measures are most powerful, which age range is most powerful, how can we get the most data out of that, how can we establish individual patient run-in data even going into a clinical trial, really as the immediate preparation for a clinical trial. And then that natural history data will be really helpful in using modern statistical methods to minimize the number of patients needed, the time needed, the placebo part needed to really make trials in an ultra-rare disease population efficient and feasible for both patient community investigators and industry to do that. And having that data will also convince industry partners really to dive into that, because we can show we can actually do this, we can measure this, we can be efficient in doing that. And so I'll leave Reghan for the final slide here.
Sure. So it's kind of highlighting what Dr. Bönnemann just said, which is basically preparing for these clinical trials and getting set up for success, really strategizing for our success as a community. And so this relies on a collaboration between affected individuals and their families, which we have a great collaboration already, between clinicians and researchers, patient organizations, academic and federal organizations, and pharmaceutical companies. And as these therapies are in development, our clinical trial readiness efforts continue to occur in parallel, so that we may enable our patient population to really be ready to benefit from the clinical trial opportunities that are coming. I think we finished in time.
Any final words from Dr. Beck, so Dr. Dziewczapolski?
I would just emphasize what Reghan just said in terms of the necessity for collaboration among different groups. I mean, I often think of an analogy of a three-legged stool, where the legs are the patient community, the academic community, biotech and pharma, and then, of course, every stool needs a seat. And I think that's what patient advocacy organizations like Cure CMD, Team Titan, and all these other great organizations that everyone belongs to really kind of bring this all together. So thank you all.
Thanks so much, everybody. This is really great.